Hello? 1, 2. Okay, thank you. Just one question. Is Chris Guilfrey in the room? Not yet. So we have lost 2 speakers. Oh, it's a lot. Since time is short, maybe we will change the order of the lectures, and that it will give more time for the latecomers. So thank you very much, ladies and gentlemen, to come to this ETPN sponsored session. So just a few words about ETPN. ETPN stands for European Technology Platform on Nanomedicine. This is a European association with 140 members coming from academia, the clinics and industry, so representing more or less the 3 pillars of nanomedicine. And our main mission is to provide the European Commission and the member states with R&D priorities, so that's to support the development, not only scientific but also industrial development of nanomedicine. And anyway, you will find all the information and deeper presentation of but European Technology Platform on Nanomedicine on the website. Just Google ETP or ETP Nanomedicine, and that's it. You will get it. And I would especially recommend you to read the white paper on nanomedicine under Horizon 2020, which summarizes, let's say, the roadmap we have, let's say, discussed with the European Commission for the development of nanomedicine in the next 7 years. So now we will have today. Okay, thank you. We have today 5 speakers. Mr. Tom Krabby from UCB Pharma had to cancel at the last minute. So the first speaker is Dr. Scott McNeil, who is the director of the Nanotechnology Characterization Lab at National Cancer Institute in Frederick, USA. And Scott McNeil will present, let's say, the Nanocharacterisation as the first milestone in translation. So, Scott, the floor is yours. Yes. So this is the. Okay. Could you have the slides of Dr. Scott McNeil? Johan Ach is later on. While the technician is looking after the presentation, just one additional word about translation. The focus of this session is on translation, and by translation we mean moving the preclinical proofs of concept to the clinic and to the patient, I mean to the market. And you will see that, of course, this is not an easy way, this is a long and chaotic way. And so we have selected the speakers who will present you one after the other, let's say, the different facets of this translation pathway from lab, or let's say from lab to the patient. I do not know anybody over there. Can we have the next? Can we have the slide set of Professor McNeil, please? Okay, please. D'accord. Oui, donc les autres. C'est Chris Guilfrey, je ne sais pas qui c'est. Je ne l'ai pas vu encore. Ah oui, ok. Sorry for the technical delay. B plan, apparently. Okay. So the slides have been lost. They are somewhere in the room, but maybe not on the right computer. But are the other presentations downloaded on the PC over there?
Well, he said he just only had the presentations.
Only one? Yeah. Oh, yes. But
they were handed in. I know that I checked with the technician before.
Okay.
So it's probably...
So they've got them, but they more or less lost them. Okay. Well, good point. Scott, are you ready to make it hand-free and... Good. Thank you.
No, that's fine. So, Patrick, thanks for the invitation to present here at Clinam, and thanks also to Clinam organizers for giving us a chance for the third time to tell you what we're doing at NCL. I'll say the same thing that I started my talk with last year. The Clinam meeting is one of the best meetings that I enjoy attending, based on the content of the discussions as well as the networking and just the amount of nanomedicine VIPs that we get to interact with here. So many of you recall, if you were here last year, that NCL had a 4-hour session to give you our lessons learned. So it was a little bit difficult for me to look at our slide deck and figure out what I was going to talk about and avoid just duplicating some of those efforts. So I do have a few, 3 or 4 slides on what NCL is for those of you that are not familiar with the Nanotechnology Characterization Laboratory. Then, as the title suggests, I'm going to discuss some of the milestones, specifically the characterization, which is our middle name, and interaction with regulatory agencies. So NCL has been in operation for 10 years this September. How time flies. We're a formal collaboration among the National Cancer Institute, NIST, and FDA. It's a very strategic interaction and productive interaction. It's not just on paper. We interface with those entities at least on a monthly basis, if not on a weekly basis. So in brief, NCL performs preclinical characterization of nanomaterials that are intended for cancer applications. When a particle is submitted to NCL, we will subject it to this 3-phase assay that you see here, the first being physical-chemical characterization, then in vitro characterization, and finally in vivo characterization. And we're not just simply a CRO that hands a data package back to the sponsor, to the client. We will defend that data in front of FDA as applicable, and in several cases, we'll also explain the data and validate it as a third-party, unbiased entity to venture capital companies that are interested in investing and doing an equity investment in some of the companies that come through NCL. Under our assay cascade, we do, as I said, PCC, where we look at size, shape, surface chemistry, stability, many of the parameters that you see up there. And then keep in mind that each one of those parameters probably has 5 to 10 different instrumentation methods and techniques to go after each one of those parameters. Under in vitro, we're primarily looking at the interaction of the biomaterials with the nanoparticles, and specifically interaction with the immune system. So we do ex vivo studies using human fresh blood to look at things like opsonization and phagocytosis, and also complement activation, which is a very big deal for nanomaterial biocompatibility. And finally, in vivo, we do ADME tox, which is very similar to what you would see in a pharma company, where we're generating data in support of the pharm tox section of the IND. We've, in 10 years, characterized over 300 nanomaterials, those that you can see up here. And historically, we understand the strengths and limitations of each. There's not one that is a panacea for any specific drug. Depending on what you want to do and the clinical indication, there's usually a best fit for that. Of course, there's many other complications for any of these, including scale-up and quality control. Here are some of our collaborators that we've had over the last 10 years. Many companies that you're probably familiar with, including many European companies, have gone through NCL. And as I said, we don't just stop there and hope that they get into clinical trials. Now 7 have progressed into clinical trials. 7 out of 300, we view that as a pretty good average. Many of these are in either phase 1 or phase 2, and we expect 2 to 3 more this upcoming year. And what's very nice in the last 18 months or 2 years, we've also seen investment by VC as well as pharma into nanomedicine, because just as anecdotally, prior to that, it was primarily universities and SMEs that were submitting material to NCL. So it's very nice, very rewarding to see pharma get involved, because without that pharma pull, it's often just a academic exercise. But now we're seeing both the interest and the investment in nanotech, where more than $1 billion were invested in nanomedicine, assuming all the options are exercised. Okay, so what are some of the challenges that we face that that I'm sure that many of you are also facing? So I'll go through a few of those, and maybe we come back to them during the question and answer session. So nanomaterials are often very complex. They're multifunctional. They've got a drug. They've got a platform in the nanoparticle itself. They've got a targeting agent and sometimes even an imaging agent. That complexity translates into complexity in scale-up and large manufacturing. So certainly we have to have a very good understanding of how those come together, the best stoichiometry, the best number of ligands, as you heard earlier today, to which is best optimized for efficacy in vivo. So I mentioned scalability. It's very nice to have a top-tier publication for a nanomaterial that has no possibility of translating into clinical trials just because it is complex. So as long as we can scale that and going from the milligram proof of concept into the gram, into the tens of grams and into the kilogram, that is not a linear process, as many of you can attest to. We want to make sure that we do have quality control, that we have an analytical method to determine, for example, how many receptors are on the surface, and whether it's important to know how that's distributed, whether we just sample the entire population, or if the regulators are going to ask, well, tell me the breakdown of that population. Are some of those nanoparticles with 0 targeting molecules and the rest have 100, and how does that work out? And also reproducibility. I'm sure many of you are aware how complicated that is, even with something as historically in use as Doxil. If you can imagine something that's even far more complex, being able to get that exactly right, batch after batch, under GMP conditions can be very complicated. A few more examples. So those were the problems, but here's how we go about doing that. The most obvious, is for physical-chemical characterization, knowing what parameters are important. So NCL has several dozen, in excess, I think, of 50 different protocols that are available, but not every particle goes through each of those 50 assays. We have to know which ones are germane to quality control and efficacy for that particular nanoparticle. So we also evaluate biodistribution. Often an investigator will approach us and say, well, we tested 6 rats with our nanoparticles, and none of them fell over dead, so therefore it's non-toxic. Well, that's just not good enough for translational sciences and regulatory approval. So we do non-GLP studies. For each data point, we have 5 to 6 animals, and we'll have multiple arms. So an animal study for us is in excess of 100 animals, and we look at 42 different tissues very rigorously to look for things like lesions in addition to gross toxicity. Something that you should be very familiar with is even small changes in a physical chemical property can dramatically affect biocompatibility, either toxicity or efficacy. We've seen that over and over and over again. So that, again, speaks to the importance of characterization in being able to translate the concept into clinical trials. What we're finding, though, is a SME may have a terrific idea, but then they're stuck with, okay, I need to go from that tens or gram amount into a kilogram amount for a clinical trial. What we found is it's difficult to locate a manufacturing organization that has the instrumentation and the quality controls to scale that up to the kilogram level. Many are eager to do it, and they'll say, yes, we can do that, that's no problem. But then if you go in depth into their analytical tool set, they have an HPLC, and that's all they have. It just simply has a UV vis detector. Well, many of these multifunctional particles need far more than that as far as instrumentation for quality control checks. Often what we find is that several of them are not interested in retooling for a small batch, one-off run of a nanoparticle. And so what happens is that the nanotech company ends up subsidizing the CRO for that initial batch. And so it's twice or thrice the burden because often they are subsidizing for instrumentation that the CRO may not have. And so there is a huge demand for one or more GMP scale-up facilities for nanomedicine. We can do the lipids fairly well, but certainly nothing is commonplace for some of the other materials, especially for some of the polymeric-based ones. So there definitely is a need for scale-up facilities, and I see that the EC and the EU are ramping up for that, and I certainly applaud that effort. Because, as many of you know, once you get past the phase 2, you're stuck with a process that you've since developed. And so you can do phase 1 or phase 2A with a small-scale batch or a small-scale facility. But then after that, you're stuck with what you choose as far as future phase 3 and final approval. So NCL, as hopefully you've picked up, is a transnational resource. We used to say national resource, but now we're certainly international. And it's a free resource for nanotech developers. Under PCC and for farm tox, and especially for the immune response, which is often something that's overlooked, because in cancer clinical trials, that's usually not mandated until phase 2. However, we can find very early, if we identify those, many of our assays are validating for in vivo responses. So we get a priori look at what we anticipate is going to happen under in vivo conditions. So under PCC, we also help with answering FDA questions that are posed during a pre-IND meeting with FDA. Oftentimes, the FDA will ask a question of the sponsor that they just simply do not know how to go after. So they will come to NCL with those questions, those series of questions, that actually, believe it or not, makes our job far easier. So rather than starting from scratch, we know exactly the type of data that's required for linkage in the IND that FDA is interested in. Same thing with farm tox. We're developing a number of assays that look at things like stability, drug release profiles, encapsulated, bound portions of the API, which are, again, very germane to efficacy and safety. I mentioned that FDA will ask questions. It's also rewarding to see that over the past 10 years, due to our interaction with FDA and our tight association with them, we offer training sessions with FDA reviewers. And so in some of that education, we would pose questions to FDA reviewers and help them understand instrumentation and measurands and parameters that are important for regulatory evaluation. Then FDA reviewers will ask those questions of the sponsors as the sponsors approach them, and then those sponsors will come back to NCL and ask us for help. So it's cyclic, but it certainly shows that the system is working. Here's an example that I'm showing you here where the FDA wanted to know the number of ligands that were on the surface of a gold-bound particle. That's a lot easier said than done. There's very few techniques that can give us an answer on a per particle basis. So what you're seeing here is the gold-based particle. Then we were able to immunolabel with gold an antibody to the ligand that was on the surface. So you can count there's roughly a dozen or so. That's not how the particle normally looks. It's normally just a single gold, but it makes a nice picture to show that you can count by EM the number of ligands that are on the surface. Another example is for a company that was asked to compare the amount of protein binding for docetaxel compared to the legacy Taxotere. We were able to help them with that. There was changes in the formulation, meaning the lipid components, and so it was a valid question, and we were able to offer some of our analytical tools towards that. Another example that were very specific questions to a company is helping a radiopharmaceutical assess whether or not the catheter was changing the formulation, if it was degrading, oxidizing, if it was anyway interfering with it. So we were able to take the catheter and the exact catheter that they were using in clinical trials, run the formulation through it, and examine it before and after for all of our physical chemical properties, including endotoxin contamination. There's many questions that we address and answer on our own without specifically being tasked to it. One is to quantitate the pegylation that's on the surface. This is now formally requested or identified as a problem by EMA because there's many different forms. It's difficult to do because the peg doesn't light up via UV-Vis techniques, and so we were able to develop a number of techniques that can now be used. We share all of our protocols on our website if this is something that interests you. So we offered 3 different techniques. We've published on the one way down at the bottom, the chemical methods, where we simply strip the particles using something like DTT and then quantitate them on various gels. Next we have CAD, or charged aerosol detection. That is shown to be fairly sensitive. One of the limitations of that, though, is you need to be able to charge the PEG species. And then TGA gives us terrific results, but it takes almost a gram of material. So make sure you have a lot of material before you approach that technique. Again, I think most of these protocols are now available on our website. Something that we are really emphasizing this year, and including previous years, is you have multiple fractions of drug of API when you're dealing with nanomaterials. So just for example, let's say I have a micelle that has an API drug inside of it. Well, I have, or better yet, a polymer-based one that is a prodrug. So I have the API that is bound in the particle itself. I should use the word encapsulated in the particle itself. When that leaks out, you also have a kinetic of that initial release as the prodrug, and then you have the cleavage of the prodrug into the active form. Okay, depending on your technique, you may lump all those together and get an erroneous measurement, when in fact the main constituent for efficacy and toxicity is going to be that, hopefully going to be that the final cleaved drug, the API itself. So we have a number of techniques to do that and look at the distribution. So we have a traditional blood partitioning assay to look and see how quickly, especially for a hydrophobic molecule, will leach out of various nanoparticles. We can label the various components. So for example, we've published on labeling both the API and the carrier and to look at stability. I don't know if you can see that from there, but we've published on it. We showed very early on, as within the first time point, that the API separated from the carrier, from the lipid carrier. Something that we're now looking into quite a bit is the last bullet there, looking at a stable isotope to measure the encapsulated versus unencapsulated versus the free versus the bound. That's where we're headed as far as our techniques. Some of the others, we didn't have the confidence that they were not interfering with the formulation itself. For example, is a liposome stable in something like a solid support technique? We're also very involved in monitoring the quality and the bioequivalence between batches, as well as if somebody puts forth an alternative to a legacy drug. So here's an example of one of the strengths of Nanotech is we can change the PK profiles of existing APIs, change the PK as well as decrease toxicity. What you're seeing here is a reformulation of tumor necrosis factor alpha. This is reformulated by Cytomune in Maryland and the USA. What I'm showing you here in red, that very small curve, is what TNF looks like if you injected IV alone with only a carrier. It has a half-life of, as you can see there, of roughly 26 minutes. By changing that formulation and attaching that same drug to a gold nanoparticle, that half-life goes up to an hour or 3 hours, 182 minutes. This, as I said, is one of the beauties of Nanotech. Is it gives new life on previous molecules that were either toxic or had very limited PK profiles. And you can see that combination of the nanoparticle-bound TNF there in green. I should say that the toxicity was dramatically changed with this new formulation as well, in that, as many of you know, in the late 1990s, TNF alpha was used and it caused lethality, caused deaths due to the immunotoxicity. But this company has been able to triple that lethal dose that was obtained back in the 1990s, again, simply by attaching it to a nanoparticle. I mentioned earlier, it's very important to make sure that you're monitoring the unencapsulated form as opposed to the total amount of drug. There are various methods to do that, but make sure that every particle is unique, but make sure that that is the portion that you're monitoring. And just briefly, I'll wrap up the difference between biosimilars, and you heard the term this morning by Garrett, nanosimilars. So there are considerations due to the product itself as well as the process. And there's no such thing as a nano-identical, but there certainly are nanosimilars. And I'll refer you to the NBCD working group that Garrett mentioned if you have an interest in that. But as you can see there in the graph, even the same company that's making Doxil in the same facility had differences that we were able to monitor between the old Doxil and the new Doxil. The new Doxil was actually less toxic as monitored by complement activation. So with that, let me recognize the true smart people at NCL. That's my team, and Neil Pottery runs chemistry, is also my deputy. Steven Stern is our toxicologist. Emina Dobrovolskaya is our immunologist. And we have a poster here, and Dr. Jennifer Grossman will man that if you have an interest in working with us. So with that, thanks for your time.
One, two. Okay. So we have time just for one or two questions. Anyway, at the end of the session, we'll have a longer question time. One, ladies first, and then Chizzy.
Scott, you started to touch it on this topic towards the end, but ultimately what's important is the amount of bioavailable drug at the site of action, right, which can be very hard to get at. Are you making any progress in being able to measure that?
We can do a standard biodistribution study, but as far as excising a tumor and looking at the API, we have not done that. We would look at overall efficacy as monitored by tumor size. So to your question, not explicitly, no.
Not a question, but a comment. I think NCL is really one of the best things that happened to nanomedicine. And I think that, everyone should try to work with this guy, and hopefully equivalent institute will be opened in Europe and other places, because I think many of the small and even many of the large company does not understand what it involved to develop a nano drug. And I think this guy, Scott, and his people really understand it extremely well. I think this is really a great achievement for the field.
Thanks, Hezi. By the way, I paid him to say that.
Okay, thank you very much once again, Scott.